Hello students and my NEET aspirants. The topic for today is cell, which I have made it very easy for you all, starting from discovery to nucleus. Let's start. Now what is cell? Structural and functional unit of life made up of protoplasm surrounded by cell membrane and capable of self-reproduction. Now let's go directly to the discovery. As we know, Anton von Leeuwenhoek first discovered blood cell under a microscope. Robert Hooke named it as cell, and he observed Koch cell under his microscope and drew a sketch in his book Micrographia like a honeycomb. Cell theory put forward by Schwann, Schleiden and Virchow. The 3 points are given, that is, all living organisms are made up of one or many cells. Cells are structural and functional unit of all living beings, and all cells originate from some pre-existing cells, cellula. Then we have Robert Brown, who first discovered nucleus of orchid leaves. Then we have types depending upon the number: unicellular, multicellular, shape, spherical, oval, cylindrical, slipper shaped, size, largest, longest, smallest. Then we'll go to the cell structure. We have cell cover and protoplasm. What is cell cover? Cell cover is cell wall in plants, as well as cell membrane is born in both plants as well as animal. Difference is very important. Cell wall is made up of cellulose, non-living, thick, rigid, freely permeable, while cell membrane is made up of lipoprotein, living, thin, elastic and semi-permeable. What about cytoplasm? Cytoplasm is the fluid part of the cell outside the nucleus. Cytoplasm has cell organelles and cell inclusions in them. What are the cell organelles? I have divided them as double membrane, single membrane and non-membrane. Double membrane is mitochondria, forms ATP in Krebs cycle. During Krebs cycle, plastids, chloroplast, chromoplast, leucoplast. Then we have single membrane structures, endoplasmic reticulum. The functions are being given, that is transport, protein synthesis, lipid and steroid synthesis. Golgi complex, which is characterized by cisternae and vesicles. Function is secretion of hormones, enzymes, proteins. Then we have lysosomes, which are suicide bags and have hydrolytic enzymes in them. Lastly, we have the non-membranous structure, that is ribosome, which functions as protein synthesis. Then we have cell inclusions. There are 3 cell inclusions: vacuoles, granules and waste products. Vacuoles, the fluid in the vacuoles is cell sap, very big in plants and very tiny in plant cells. The covering of this vacuole is tonoplast. Granules are nothing but reserve food of many types. Waste products is latex in rubber plants, tannin in tea leaves, caffeine in coffee, asafoetida, etc. Lastly, we come to nucleus. Nucleus has 4 parts: nuclear membrane, nucleoplasm, chromatin material which changes into chromosomes, has DNA, and nucleolus which has RNA and functions as protein synthesis. I have given 2 extra points, that is, some living beings have 2 nucleus, as in paramecium it is absent. Nucleus is absent in RBCs and sieve cells, while nucleoli are 2 in number in liver cells as well as in some lymphocytes. That is all, kids. I hope it helps.